Hello students, now we are going to discuss about the historical development of microbiology, right? So in 5 minutes I will tell you the scientist name right from the 12th century to till today, right? What is their contribution, in which year they have done, and what for they are known for, right? So that if you will come to know in summarized way, it will be easy to remember in exam point of view. So let's say start historical development of microbiology. The first name which comes in is the Antony van Leeuwenhoek's, right? 1632 to 1723. He told about the microbes nature and how the microbes will be there in their particular nature in their invisible environment, and he told about lens also, like with the proper lens and with the proper a piece of drop of water we can see the microorganism in the magnified way, or any glass piece can be used to see the small organism into their habitat. That was told. The second thing, Louise Pasteur, very famous name, students. It is without Louise Pasteur name we cannot imagine the microbiology. In 1822 to 1895 he told about certain important theory like pasteurization, like pasteurization, like that we can correlate. Pasteurization is the process of the pasteurization to the milk, right? Whenever we are making the bacteria, living organisms into the milk, into their inactivated state, so that it will not be spoiled, right? Then fermentation, second important theory. Fermentation is very important, which will be done by the microorganism into the big bioreactors for the production of antibiotic, production of alcohol, production of so many other beverage, right? And that's what, because of this important contribution, he is known for the father of microbiology. The third one, Robert Koch, very important name, right? 1843 to 1910. He is also known as father of modern microbiology. Louis Pasteur is father of microbiology, but Robert Koch is father of modern microbiology. He told about the bacilli, which caused the tuberculosis, and cholera bacilli also he told about. He told about the solid media also, right? Then coming to this other scientist, which is without them their contribution microbiology is incomplete, right? So coming to here, Roger Bacon, right? Roger Bacon, 1220 to 1292, in that century he told about disease is caused by invisible microorganism. कोई भी any disease, it is causing by the invisible or not visible microbes, which may be there in the form of anything, right? Then the next scientist, Girolamo Fracastoro, right? 1483 to 1553, right? He told he has also done the similar types of observation, that he also told the invisible microorganisms are causing the different types of disease and disorder. Then later on he given the germ theory, right? Fracastoro given the germ theory, right? Then that's what Girolamo, G germs, zero, right? So like that you can correlate. Then next, Athanasius Kircher, 1601 to 1680. He told the worms are invisible to naked eye. To see the worms we need to have the durbin, we need to have the microscope, we need to have the specs, lens, so many things, but these are invisible in the naked eye. That was told in the 16th century, just imagine, right? Then next, John Needham, 1713 to 1781. He told the raw meat, if you will expose into the normal room temperature, organisms will develop. So that was also one of the good achievement at that time. Then Lazzaro Spallanzani, 1729 to 1799. He did one small practical on beef broth, right? Beef broth, he closed tightly beef broth, and he kept it for incubation. After incubation there was no microorganism. It means if any bacterial attractant, maybe beef or meat, if you will tight with the proper airtight or cotton or aluminum foil or whatever it is, there will be no microbe. Means the microbes cannot transfer from the inside to outside of the broth, and outside to inside of the broth. So that was the also very good discovery. Then Franz Schulze and Theodore, 1810 to 1882. If air is passed by strong acid solution, there will be no microorganism. Means if any air, right, any air, it is coming to the room, or it is going to the laminar airflow, it will be passed through strong acid solution. So acid is going to kill them, and whatever the air we will get, it will be aseptic air or free from microorganism. The air will be free from microorganism. That was told by the Theodore and Franz. Then later on, H. Schroeder and T. Van Dutch, 1850. What they told is bacterial culture tubes with cotton plug, right? Any bacterial culture tube, right? If it is with the cotton plug, there will be no microorganism. Microbes cannot enter inside and they cannot go outside. That was told by the Dutch and his friend, right? Coming to this side, pharmaceutical microbiology contributions by the different scientists, discoveries, right? So the first name come under this Edwin Klebs, 1883. He told the diphtheria bacillus, about diphtheria which is causing by diphtheria bacillus, this name bacteria, that was discovered by Edwin. Then Hansen, 1874, he told about Mycobacterium leprae which is causing the leprosy. Then Neisser, 1879, Neisseria gonorrhea, gonorrhea is caused by Neisseria gonorrhea, was discovered by Neisser. Then Augustin, 1881, he told about Staphylococcus aureus which is causing the wound infection. Then Nicolare, 1885, Clostridium tetani, he told about Clostridium tetani which is causing the tetanus. That's what, after getting injury we are getting TT injection to reduce this infection, right? Then Siga, SIGA, 1898, okay. Then Shigella dysenteria which is causing dysentery. Then Saudin, 1906, he told about Treponema pallidum which is causing the syphilis, which is reproductive organ disease. Then Bordet, 1906, he told about the whooping cough. So you can see that lots of bacteria and microorganisms, it has been identified right from the 18th century to 19th century, and that's what we are having today. Different types of antimicrobial agent, different types of antifungal, antiviral, and so on, right? We are having different microscope, we are having different types of sterilization techniques. These all are because of our scientists in the right from the 12th century to till today. They have identified, they have given different theory and concepts which made it is easy, right? Thank you.